So Woody Hastings was interested, still is interested in bioluminescence. And so historically, half the lab worked on luminescent bacteria and half on dinoflagellates. And that's how Woody and V.C. Sweeney got into rhythms. Woody's lab cycled in terms of emphasis, depending on probably random factors. When I got there in 1974, nearly the entire lab was working on bacteria, and there were only a few people interested in clocks. And over the next 5 years, then it switched over, so that by the time I left, almost everyone was working on clocks, and no one was working on, few people were working on bacteria anymore.
So you went there as a postdoc or to do your PhD?
To do my PhD. Intending to...
As a biochemist?
As an ecologist, more or less. I have an undergraduate degree in degrees in oceanography and chemistry, and I was going to be a biological oceanographer. I was going to go to graduate school at Scripps in San Diego, and sort of on a whim at the last minute decided to study, learn physiology and biochemistry on marine organism. So everyone doing biological oceanography was and is asking the same questions: who's there, what are they doing, and how fast are they doing it? I thought to answer the latter questions, it would be good to know some biochemistry. And I met Woody, and we got along well. And so I thought, well, I'll go to Harvard, learn some biochemistry for a few years, and get back into biological oceanography. So at Harvard, I actually taught oceanography for 3 years, but I was simply seduced by clocks, which I remember walking into the biolabs between the rhinoceri and thinking to myself, this was simply a great problem, and no one is asking the right questions. The Nobel Prize is nowhere in sight, and so this is a field that one could move into and actually do something of significance. It's not solved, so I ended up picking up clocks as a problem to work on. Mm-hmm. And Woody's lab was a great place to start that, because it was just there were lots of resources and very few constraints. If you could think of a good experiment to do, you could just go ahead and do it. All the rope you need to hang yourself. So people said, that the best and worst students to come out of Harvard came out of Woody's lab. So this is in the mid-1970s, and at that point there were many physiological rhythms known, but there was no known case of an enzyme whose activity was actually controlled by the circadian clock. And I thought luciferase in goniolax would be a good first case for this. So I purified, so one set of experiments was strictly biochemical. I purified the luciferase to more or less homogeneity, enough to make antisera, and proved that the clock was regulating bioluminescence by regulating the synthesis and destruction of the luciferase. So there's actually more of the same enzyme there at night than there was during the day. And I purified the substrate and worked with Osamu Shimomura at Princeton to determine the structure of that. Which is another sort of interesting story. It's a linear tetraperol that gets excited by oxidation to emit light. So it's sort of the physical chemical reversal of photosynthesis. So you have a circular tetraperol that absorbs light and makes chemical energy. And for bioluminescence, you break that circle. Now you have a linear tetraperol, and you excite it, and it emits light. So it's some fun cosmic unity, as it were. And the other set of experiments were clock organized, that is, having gotten interested in clocks. Again, this is in the pre-molecular era, and there was at that point a lot of interest in determining what part of cell physiology was involved in rhythms. Biological clocks were notoriously not affected by inhibitors, with just a few exceptions. And one of the ones that emerged while I was there, and that we had a lot to do with studying, were inhibitors of protein synthesis on ADS ribosomes. And I think these are still good experiments, although they've largely been forgotten because you can't get the papers by PubMed. So no one under 40 has read them. But the observation in a number of microbial systems, and later extended to pinealocytes and some vertebrate systems, are that Gene Block did some nice work in bulla. You have to make proteins on ADS ribosomes for the clock to run, and that's perhaps, you say, well, gee whiz, that's not very surprising at all. But the interesting thing is that there are times of day when you have to make the proteins. So if you simply slow down, if you stop protein synthesis, the clock stops. If you slow down protein synthesis, you can slow down the clock. And you can reset the clock with pulses of inhibitors. So that was what I think was, to many of us, was very interesting. So it's not simply that you have to have proteins there for the clock to run. It's that a part of the timing cycle requires synthesis of proteins at certain times of day. And it's not only true in goniolacs and bulla and pinealocytes, it's in acetabularia, it's true in acetabularia that don't have a nucleus. It's not simply that the proteins are required, it's that synthesis of ADS, synthesis of proteins on eukaryotic ribosomes has to happen at a certain time of day for the clock to run. So this is something you hang your hat on. So it wasn't Golgi function. It wasn't proteins coming apart and forming complexes in the cell membrane. It wasn't cyclic AMP and phosphodiesterases. If you look at the history of molecular models, the 60s and 70s were the era of when a lot of biochemistry began to come together. People's view of membranes was revolutionized. Cyclic AMP was discovered, and signaling was discovered, and so any time there was a new insight into biochemistry, 6 months later there was a model for the circadian clock based on this, and they were all plausible. If you selected some data, you could generate a feedback loop, but they weren't compelling. And I think that's still how I view a lot of modeling, modeling that isn't tied to biochemistry. It's not hard to make models that are plausible, but it is hard to make models that are compelling. So that's why these protein synthesis inhibitor experiments were compelling in that they really pointed now to a part of biochemistry, a part of cell metabolism that had to happen for clocks to run.
And is that about the time when the idea came up of feedback loops? When was that?
Well, they originated with Goodwin, of course, in the 1960s, and in a lot of ways the mathematical basis for the transcription translation oscillators is all there in Goodwin's work. It's just that it was not compelling to me any more than the Cummings cyclic AMP model or the new Saltzman-Hastings membrane model or any of the other models. They were plausible, but not compelling.
What
So,
did it need?
Well, it needed more data and more testing. That's right. So the late 1970s was the time at which recombinant DNA technology was created. this offered the possibility of actually doing the right experiments. So I was casting about for postdocs for what to do, and I decided to work on clocks. And I realized that if you're doing biochemistry on clocks, you're really working in the dark because you don't know. You're hoping for a lucky guess. You're going to suddenly make the lucky guess that this is the important protein, and so I'll study that protein. And if you're not lucky, then you're SOL. So in order to know what to study, you really need to do genetics. Because the beauty of genetics is you don't need to know anything. You need to have an organism that does the phenotype, has the phenotype you're interested in, and that has sex. And you can make mutants. And if you can do those things, the organism will tell you what you need to know. It will identify for you the genes that are pertinent for the phenomenon.
How cunning.
So it's exactly the right thing to do. So in the late 70s, genetics had been applied to rhythms in two organisms. In Drosophila, both at about almost exactly the same time in the same place. Drosophila and Neurospora, both at Caltech, both in the early 70s, in close to adjacent labs by Ron Konopka and Jerry Feldman. Konopka's work has gotten more play, but Feldman was equally, I think, a pioneer in this. Konopka published first, but the experiments were really begun about the same time. So by the late 1970s, there were mutants in flies, and there were mutants in Neurospora. Each had identified a single gene, period, and frequency, respectively. In the late 1970s, you could transform Neurospora, and you couldn't transform flies yet. Biochemical genetics had been invented in Neurospora. That is the association of single genes with single proteins and metabolic pathways. And it seemed to me then, as it does now, that the answer, if you want to ask the question, how does a clock work? The answer you want to give to that is in terms of genes and proteins. And so to answer those questions, you want to be able to manipulate genes and proteins in a biological context, in a cell or a tissue, whatever, an organism, where you can manipulate them. So to do that, you have to be able to transform. You have to be able to manipulate genes, put them back in the organism, and watch them perform. And the organism where you could do that was Neurospora. You couldn't do the experiments in flies,
Yeah.
although subsequently, of course, you could. And it's turned into a marvelous system, as Jeff Hall and colleagues at Brandeis have shown.
So then, you went to Feldman's then.
So there was Feldman, and I went there I went around the world for 6 months, which was a wonderful experience. And then I went to Feldman's lab supported by a Damon Runyon Fellowship in the early 1980. Charlie Yanofsky's lab was at Stanford, and that was one of the places where the Neurospora technology was very well known. The recombinant DNA technology. Jerry was going to do a sabbatical there and learn how to do recombinant DNA. He did do the sabbatical, but I guess he and Charlie didn't get along or something, but Jerry didn't pick up the technology. So I did other experiments for about 2 years. Did a series of sort of an extension of the work I'd done at Harvard, but taking advantage of the Neurospora strains. So I did phase-shifting experiments with pulses of cyclohexamide, an inhibitor of ADS protein synthesis, in the long and short period mutants of Neurospora, which was really sort of fishing just to sort of get my feel of the system. And to my surprise, I found that you could get Hideaki Nakashima had shown in Jerry's lab that you would get a phase response curve for pulses of cyclohexamide. So there was, like in every other organism, there was a sensitive phase and a non-sensitive phase. What I found was that where 3-hour pulses of cyclohexamide gave nice big phase shifts in wild type, they had almost no effect in long period mutant. So that's a surprise. But longer pulses would have an effect, and you got it wasn't just pulses. I also did dose response curves and showed that they were really less sensitive. Sensitive at the same circadian time, but much less sensitive. So I made a model. So this reinforced in my mind the idea that proteins did have to be made at certain times, and we hypothesized that maybe the nature of the mutant protein in these long period mutants was that the protein is more stable, so that short duration inhibition had less effect on concentration. That's turned out in the fullness of time to be correct. So we did those experiments, and then by that time it was clear that as it had been when I went there to clone frequency. Went there in 1980, and I wasn't going to learn it in the Feldman lab. So in order to pick up the recombinant DNA technology, I went next door into Harry Noller's lab, who's it was an RNA lab, and Harry was the person that one of the people that crystallized the ribosome and did its structure. And at that point, they really knew how to do this stuff. So a technician there, Joanne Kopp, taught me how to do DNA, run gels, do restriction enzymes, clone things. So it was hard to do in those days. Make libraries, and so I learned to do it there. I did not clone Freak, obviously, in Santa Cruz. It was a very small lab. There was one other person in the lab that was really smart, and that was another. She was a graduate student, and we coexisted for, I guess, probably 2 years, sort of checking each other out. As Jennifer has subsequently said, familiarity breeds attempt, and so after a period of time, I attempted, and we got to know each other, and subsequently, of course, we got married. And Jennifer and I have worked together since then. Looked for a job in. Jerry and I also did not get along, in truth, and so I was anxious to leave and stay there as long as I did because Jennifer was there. We couldn't wait to get out, and started looking for a job in '82. And struck out completely. Looked again in '83 and got a single job offer. Clocks were not hot then at all, but I got a job offer at Dartmouth in the medical school in biochemistry. It was a pretty good offer, and in fact it was the best offer I got. But it wasn't where I wanted to go, but I went there in early '84 and started to set up a lab. Jennifer finished her graduate work later that year and joined me in the fall. We were married in September 1st, 1984, and she came and we drove out from California to Hanover in September. So ever since then, we've been there. Jennifer focused on output from the clock-controlled genes. That was her work, and I focused on how the oscillator works. So I tried several things. Finally, settled on doing a chromosome walk to get to Freak. And I started that, and partway through that work recruited a postdoc, Rob McClung, and Rob and technician Barb Fox and I did the chromosome walk. We cloned Freak in early 1986. Per was cloned in 84. We published it. The bar for what to publish when you clone something increased very rapidly through those years. If we had cloned Freak in 85, we could have published it right away. Cloning it in 86, in order to get the paper I wanted to get, we kept having to do more and more things. So we eventually published it anyway in 89, but we did get it on the cover of Nature. So that was where I thought it belonged, someplace like that. The same year, I almost didn't get promoted. I will say I didn't publish any papers for 5 years after taking a job. But I had a pretty good beat on what I thought needed to be done. So during the 5 years, we set the groundwork for doing molecular biology on clocks in Neurospora, and both in the context of how does the clock work and also in clock-controlled genes. So in 1989, we published Cloning a Freak in Nature, and we published the identification analysis of clock-controlled genes, coining the term clock-controlled genes in a paper in Science. So at this point then, Per and Freak were cloned. We had, and we knew that mutations in those genes affected the clock, and a plausible model was that they were in the feedback loop, and that was the plausible model that came out of the Brandeis group. But it seemed to me it wasn't airtight, that one could have mutations in genes that were clock regulated, and that through output, and that fed back to input to affect period length, but that weren't absolutely a part of the oscillator. And I think subsequently there are examples of this, particularly if you're only looking at output, for instance, behavior. We know, for instance, now that there are genes that you can mutate that will affect period length through feeding back, but aren't actually required for the oscillator, aren't a part of the oscillator. So the right experiment to do then is to start to manipulate these molecules, and we were the first to do this. So the experiment, seemed to me the experiment to do was to, if you think that rhythmic expression of a clock gene is essential for the clock, then the thing to do is to drive expression to a high level and hold it there, and the clock should stop, and then release the high level expression, the clock should start again, and it should assume the phase from the point of release. So those are the experiments that Ben Aronson did in my lab. In the early 90s, we published that in the cover of Science in 1994, and that established then that transcription negative feedback was essential for the clock to run. And from there on, it was really filling in the arrows. We went on in 1995 to show how resetting works, that light induces the expression of Freak. And if you have cyclical expression that's a core part of the clock, and you suddenly raise the level of a clock gene and release it, then that should reset the clock. And it was Sue Crossthwaite's work, and that's still the model for how resetting works, not only in Neurospora but in mammalian cells as well. We showed that in collaboration with Toshio Kimura in '97, that light induces Per, and Per then resets the clock. It was a great problem. It's a great field. I think in truth it's peaked. Fields have their heyday. In the 1980s, it was cell cycles. and the cell cycle was cracked, and now we understand how it works, and it's applying that knowledge to understand things like cancer. For clocks, it was the 90s, the late 80s and 90s. We started off with some good ideas for what to do as a field. We did them. We reduced it to the molecular level, and that is now a firm basis on which to move to tissues and move to the whole organism.
You think it's all over now?
Oh no, no, it's not all over.
Moving to another
It's
domain.
moving to a level of another level of complexity, but it's no longer phenomenology. I think that now every experiment that is done refers back to how we think we understand how the clock works, and so that, in terms of picking a problem, in terms of how interesting a problem is, it's been a great ride. It'll be an interesting story for someone in looking at the history of science. Of course, the approach that cracked how clocks work was genetical, not physiological, and genetical approaches were not favored by most of the people working on clocks. I think it is significant that the two people that did the first experiments were both really drummed out of the field by the people in power. Of the people that solved the molecular basis, many of them came in from outside the field. Mike Young, Jeff Hall, Michael Rosbash, all came in, all had credentials outside of rhythms and came in. There were really very few people that were successful that grew up in the context of clocks. One of the things I did want to talk about was how Jennifer and I worked together over the years, which we developed some ways of coping and generally, which is how it worked. She came to Dartmouth when, as a postdoc, when I had a faculty position, and cast about some for a postdoc to do, and there wasn't anything that interested her as much as working on clocks, and so she ended up doing a postdoc in my lab, which of course is always often contraindicated. She of course did very well on that. The postdoctoral work she did was the first systematic isolation of genes regulated by a circadian clock, and that gave rise to the clock-controlled genes, coining that word, and that was published in Science. And she used that then as a launch into a research track position, and got and maintained her own funding. And we published separately some, although together mostly, but separate enough to maintain a separate identity for her. Early on, we decided, though, that it was essential to co-author all the publications. The problem is, if you don't do that, then some projects become one person's and others become another person, and it's inevitable that you're going to have fights over who gets to do what. And the only way to get that off the table is to just agree beforehand that everybody's name goes on everything. And that's something we learned from Jeff Hall, which is how he and Michael worked together successfully for many years. In the early 90s, so the questions we wanted to answer were all about cellular clocks. How do you build a clock? What are the components? How do you build a feedback loop that generates time? How can you entrain that feedback loop to environmental signals so that internal time corresponds to real-world time? And once you have a synchronizable clock, how do you export time information into the rest of the cell? And those are the three questions that we wanted to answer in terms of genes and proteins. And to, in the process of doing that, explain the classic clock properties. How do you get a phase response curve out of a molecular feedback loop in light? How do you get persistence? How do you get the long time constant at 24 hours? So we did the Aronson et al. Work that came out in Science in 1994, driving freak expression high with a regulatable promoter and showing that, in fact, high freak negatively regulated itself. So there was a negative feedback loop, and that negative feedback loop set the phase of the overt rhythm of the clock. That is, if you artificially express freak and you can follow the expressed one separately from the endogenous one, you can show that overexpression of freak negatively regulates itself. And then when you release the overexpression, the phase of the overt rhythm after that is determined completely by the phase at which you wash out the inducer. So that meant that we really were in the feedback loop. We were interested then, sort of like falling off a log, to ask whether there were environmental effects. So Sue Crosswaite picked that up, showed that there was light induction of freak. But it wasn't until we actually, and I remember this distinctly, plotting the data in my office, sitting there. It's one of those eureka moments that you only get 2 or 3 times maybe in a career. Plotting the daily cycle of expression of frequency and looking at the results of light induction. That is, at any time of day when you turn on the lights, you get high level expression of freak, and realizing that that was sufficient to explain the phase response curve. If freak is slowly on its way up and the organism sees light, you rapidly go to peak. That's a phase advance. If freak is on its way down, it sees light, it goes to peak. That's a delay. Or did I get that backwards? I got that backwards. But you can explain almost completely the phase response curve simply from understanding the dynamics of the system and the fact that freak is light induced. And the same thing obtains, we showed with Okamura's lab, Shigeyoshi et al. In '97, the same thing basically obtains in mammals. So that was really fun. So that was '95. From there, it was a short step to asking what proteins are required for light induction of freak. There were a lot of suggestions in Neurospora. The white collar 1 and white collar 2 were photoreceptors. At least they were required to transduce the light signal. So the right experiment to do was to ask whether white collar 1 and white collar 2 were required for light induction of freak. So Sue Crosswaite continued her light work and started doing those experiments. And there was a complication. That is that when we actually looked at white collar mutants, they were arrhythmic. And so we said, well, all right, that's because they can't see the light. We're trying to use light cues to synchronize the clock. They're blind, and so they're not really arrhythmic. They're just asynchronous. So to get around that, we tried temperature cycles, and this is another one of the big surprises with temperature steps and cycles. They were also arrhythmic. So it was not the case that they couldn't see the light. It was that they didn't have a clock. So this was now published in May of 1997, showing that white collar 1 and white collar 2 were the transcription factors, the positive elements in the clock loop. And now we can start to put together the whole cycle. That freak would negatively regulate itself by negatively regulating its activator, white collar 1, white collar 2. And if freak cycled an amount, as we had shown that it did, then the amount of negative influence on the white collars would be cyclical. So if freak was made and then turned over, that would be sufficient to drive a rhythm. And it was at that point that we really felt that we were starting to put together the whole loop. So Martha asked the question of whether you could break the cycle into an activating phase and a repressing phase. So the experiment was to drive freak expression very high, turn off the inducer, and ask how long it took freak to decay and for levels of expression to return to normal. And then the converse experiment, starting low and going high, starting high and going low. And what she found was that it took about 6 hours from point of induction to get to repression, full repression, and it took about 14 hours to relieve repression. This is all simply by manipulating freak. And so 6 plus 14 was very close to the 21 hours of wild type. So what that said was, everyone's initial guess was that the circadian feedback loop would be a series of steps, that freak would act on something else, which would act on something else, which would act on something else, which would eventually act back on freak. And so it's just filling in the arrows. And what the white collar experiments did, and what Martha's experiment did, was said that there weren't all these steps, that it was freak acting on white collar and white collar acting on freak. And so that was the model, and that's pretty much been the case. So as we know, white collar 1 and white collar 2 interact by pass domains. They make a heterodimer. And that was the first example of a pass-pass heterodimeric transcription factor driving expression of a negative element in a circadian feedback loop. 2 weeks after we published that in Science, Joe Takahashi published Clock, and later in the year BMEL came out. That was put together then as, again, a pass-pass heterodimeric transcriptional activator driving expression of negative elements in a clock loop. So it was really 1998 when the whole picture came together. I wrote a perspective for Science that year, and End in the Beginning, by which I meant that that was, for me at least, the end of the first phase of circadian research, because at that point then we really understood how basically how the feedback loop worked, and there would be a lot of details, but it was just fitting it in.
That was the exploding 90s.
That was the exploding 90s, right?
And now there's a different phase coming in.
Right. So now is all the payback, where the taxpayers really get the fruits of this. Oh, I think they will. You asked me yesterday whether I thought the field had peaked, and in terms of the big journals, the Cell Science Nature, it probably has. But I think in terms of the important take-homes, the uses of clocks, really what we've done is laid the groundwork now so that 50 years from now, the reason this will all have been important are the things that are going on now. It was a very exciting time to be working on the problem, and it was difficult sometimes working on the center because science being inherently competitive, you don't have to think about it very hard to realize that you were aiding and abetting the people that were going to be publishing the things that you wanted to be doing.
Still did it.
But it was the right thing to do, and I don't think anyone who did it has any regrets. It was absolutely the right thing to do for the field, and it helped, it raised all boats.